Thank you everyone for coming to hear me talk about my work and the wonderful introduction, and Oxford for flying me out here from England to talk about this. So I'm going to be talking about the wonderful world of RNA modifications to kind of mix it up from the DNA talks today. And so RNA modifications are chemical alterations to the canonical nucleotides, and these change the function and the regulation of how the RNA behaves. They can be internal or external, and they're found on virtually every RNA that we've ever found. And these RNA modifications are involved in practically every function and regulatory action of RNA molecules. So they're involved in microRNA processing and maturation. They can be deposited co-transcriptionally, post-transcriptionally. They can be deposited in the nucleus and in the cytosol. They're involved in translation, folding, and proper degradation and half-life of microRNA or mRNAs. And so they're really, really important in understanding the precise function of what they're doing for the cell. And there's a lot of them. So to date, there's been about 170 RNA modifications identified through all three branches of life, with a subset that have been found in all three. And then in mammalian messenger RNA, there's about 13 of them. And so you can't have one technology with previous technologies detect all of these simultaneously. Most of them have been identified using techniques like mass spectrometry, which is highly specific, but it's really expensive and difficult to set these up, and you need really specialized equipment and analysis tools to detect the RNA modifications in a sequence-specific context. There's also next-generation sequencing-based methods, but you need a specific assay for a specific modification. And these, either you use an antibody to pull down on the modification of interest, or you use some sort of chemical that interacts specifically with the modification of interest. And so that can give you a signal, like it causes a reverse transcription stop when you convert to cDNA. So you look for your stop sites in the data. Or the modification causes a systematic miscall because the polymerase, as it copies in from the RNA to the cDNA, it misincorporates the wrong base across the modification systematically. And so with nanopore sequencing, with direct RNA, we don't do that cDNA conversion. And so the modifications are left intact on the strands. And when we sequence, the RNA modifications cause an alteration to the ionic current relative to the canonical nucleotide. So what I'm showing here is in red is an E. coli 16S ribosomal RNA that had the enzyme that writes the M7G at position 527 deleted from the genome. And then in blue is wild type E. coli sequencing the 16S ribosomal RNA. And so the ionic current at that position 527 is significantly different between the knockout and the wild type, but upstream and downstream they more or less land on top of each other. So the ionic current, we can use that signal to find the positions of modifications. And this is where the software Nanocompore comes in, where we take the ionic current at every position and do these pairwise comparisons between two samples. So briefly how this works is we have a wild type sample and then a sample that has reduced amounts of the modification. So in this example, we did a knockdown on the M6A writer. And so here we have 5 reads from each sample, and in the wild type, 4 are methylated and 1 is not, and the knockdown, 1 is methylated and 4 is not. And so when we compare the ionic current at a position where there isn't the modification in a 2-dimensional space, looking at the ionic current versus the dwell time, which is how long that nucleotide stays in the sensitive region of the nanopore. Where the modification doesn't exist from both samples, we expect these to cluster in the same space in this 2-dimensional space. And so we get one cluster with this Gaussian mixture modeling. We don't have a modification here. But instead, when we cluster the data at the site of the modification, they can cluster into 2 clusters in one or both of the dimensions. And then we do logistical regression on the original sample labels, and that gives us a p-value for identifying the modification at that site. And so we can generate these samples and make these comparisons in a couple of different ways. So what most people have been doing up to date is you have a wild type sample and then a writer enzyme disruption, either through a knockout, a knockdown, or chemical inhibitor of that enzyme. Now with this, you have to have a non-lethal knockout or knockdown, or the chemical inhibition has to be non-lethal, and it is 2 samples that are cultured independently. And so there can be some biological variability that is independent of the knockout or disruption that you do. Instead, you can also do something called in vitro transcription, which is where you make a synthetic version of the transcriptome in a test tube. And so this uses the 4 canonical nucleotides, so there are no modifications in the sample. And the advantage here is that it only is derived from a single sample. And so we can detect all the modifications at the same time. And so here's a quick diagram of how this is done. We isolate total RNA and poly-A RNA, and then send some of it for direct RNA sequencing and skip the IVT synthesis process. And then with the other half, we do reverse transcription with a template switching oligo to add a T7 promoter to the 5' end of every molecule, and then use that T7 promoter to synthesize the RNA with the T7 polymerase. And then we polyadenylate to make sure that everything can be sequenced. And so when we do these comparisons with a wild type versus our IVT sample, we got about 10 million versus about 5 million reads from each of our 2 samples. We tested almost 3 million positions, and we found 26,000 significant sites. And if we look at the distribution across all transcripts, we see a peak right at the stop codon, which is consistent with what's known about M6A. So we think that most of our signal is coming from M6A, which is expected. And then when we compare to databases of these RNA modifications, we have fairly good concordance there. And so some of the interesting things that we can do now is start looking at modifications in close proximity to each other. And so this is an example where we have an M6A site that's next to an M5C site in the 2nd exon of this gene KRT-CAP2. And then we have another M6A site in the terminal exon. And when we look at the expected versus the observed counts of these RNA modifications at single molecules for the different possible combinations that we expect on these reads, we find that site 1 and site 2, those ones next to each other, are enriched when they are the same type. So if they're both modified or they're both not modified. But when you have mixed, that they're basically under what you'd expect. And so this suggests to us that this M5C site and this M6A site are linked in some way, and they're codependent on each other. And so when we look across the entire transcriptome for M6A sites that are in proximity to M5C sites, we see that there is this enrichment around within 25 nucleotides. If we look at the distance from an M6A site to the closest M5C site, we're finding that about 12% of our data, 11.5% of our data, show that there's an M5C within 25 nucleotides of an M6A on the same transcripts. And so this is giving us a lot of thoughts that there might be more coordination between these modifications than previously identified in the literature. And so just to wrap up, the IVT sample can be useful for detecting more than one type of RNA modification simultaneously. But analysis of this data is a little bit challenging because we only know the position and not the identity. So we have to use orthogonal data in order to infer the identity of these modifications, like the sequence context or other annotations. And then we're starting to see in the last 2 years, there have been more and more publications coming out showing that there is more M5C in mRNA than previously thought. And with 2 papers that I've seen recently are showing that there is some coordination between these modifications, and they do have a biological effect. And we are planning to follow up these with experimental validation, and we're trying to get the preprint out quickly. So with that, none of this work would be possible without the wonderful people I've had the opportunity to work with. My group in Italy, the Nicasio lab, my group in the UK with the Burney lab, and then our collaborators, the Paleozola lab, and all the sequencing was done in Genoa and at Human Technopole.